Hello everyone, and welcome back to the MCAT Prep Pod Biology Review. I'm your host Mark, and today we're going to wrap up our cellular section of biology with mitosis and meiosis. This is a crucial topic for the MCAT, and it's important for biological growth, diversity, proliferation. In healthcare, mitosis and meiosis touch on genetic health, disease, and even cancer. So, in this episode, we'll talk about diploid versus haploid cells, we'll review the cell cycle, and we'll talk through the phases of mitosis. Then we'll tackle the two divisions of meiosis, highlighting how the genetic diversity is generated. We're going to explicitly compare the two processes, and we're going to talk about what happens when errors occur. So, if you haven't had a chance, please check out the website, reviewmcat.com, or you can email me if you have any questions, mark@reviewmcat.com. Get a copy of the show notes if you haven't got them yet. They have lots of good figures in it, and I'll probably be referencing some of these figures as we go along. So it's definitely useful to look at some of these images. I know you're probably doing something else, you don't have your computer in front of you, but when you get a chance, check them out. Anyway, let's dive right into the material. So, generally speaking, you have two main types of cells in your body when we talk about chromosome number. There are diploid cells and there are haploid cells. And most of the cells that make up all your body tissues and organs are diploid cells. And these are written or abbreviated as 2N. Di meaning two, and the lowercase n stands for a chromosome set. So these have two complete chromosome sets. One was inherited from your genetic mother, and the other one would be inherited from your genetic father. In humans, that totals 46 chromosomes, which are divided into 23 pairs, and pretty much all the cells in your body, except for sperm, eggs, and mature red blood cells, are going to be diploid. These are also called somatic cells. The other cell type is called haploid. Haploid meaning half, and it's abbreviated as N or 1N. That's a lowercase n. And these cells have only one complete set of chromosomes, which is half the number of chromosomes of diploid cells. So for humans, that means 23 single chromosomes. The only haploid cells in your body are the germ cells, and these are the product of meiosis, and they become what are called the gametes. These are sperm in males and eggs or ova in females. This difference in chromosome number is necessary for successful reproduction, and it's key in understanding the differences between meiosis and mitosis. So let's move on to the cell cycle here and interphase. So most of the cells in the human body are non-dividing cells, and at any given time there's probably about less than 1% of cells in an adult that are undergoing any kind of division. Of course, it varies by the type of tissue that you're looking at, but your average cell doesn't divide, it doesn't undergo mitosis or meiosis, and it's really mostly just stem cells and other progenitor cells that are primary drivers of mitosis and meiosis, because that's basically what their job is to make new cells and replenish dead cells. So before a cell divides, whether undergoing mitosis or starting meiosis, it goes through a preparatory period called the cell cycle. And most of a cell's life cycle is spent in what's called interphase, and this is the time between divisions. You can think of interphase as the cell growing, doing its job, getting ready, and that's interphase. It's just the time between actual divisions. If it's going to divide, the process is broken down into 3 stages. So these 3 stages are considered stages of interphase. The first stage of interphase is G1 or gap 1, and this is a major growth phase. So you can think of G for growth. The cell gets bigger, it makes more organelles, and it carries out normal metabolic functions. Towards the end of G1, there's an important checkpoint, and that's called the G1S checkpoint. And this is sometimes called a restriction point. So here the cell will check if conditions are right and if the DNA is undamaged. And there are key proteins like P53, which is considered a tumor suppressor gene, which we'll get into later, play a crucial role here in making sure that the cell is able to divide properly. If the cell gets the go-ahead, it's usually committed to dividing. Some cells, rather than going on to division, they might enter a resting state called G0 from the G1 state if they aren't a cell that's going to actively divide. And as we mentioned, 99% of your cells are in this G0 state. cells like mature nerve cells, mature tissue cells, etc. So most cells still go through G1, then they go to G0, and they're just part of your body. But if it's a dividing cell, like some of these progenitor cells or stem cells, they're going to enter G1 after G0. But if it's a dividing cell, it's going to continue, and it's not going to go straight into G0 and stay there. A dividing cell that passes the G1S checkpoint is going to enter the second stage, and that's called S stage. S for synthesis. So this is the synthesis stage. And the big thing that happens during the synthesis stage is DNA replication. So the cell will duplicate its entire genome. Every chromosome, which is initially single, gets copied, and it results in 2 identical copies of each chromosome, which are now called sister chromatids. And those are going to be joined at the centromere. Even though the DNA content doubles since it's replicated, the ploidy level hasn't changed. So the cell is still considered diploid, which is 2N. It just has replicated chromosomes. So we count chromosomes by the number of centromeres, and the term chromosome can be tricky here because it can mean the single structure before S phase or the duplicated structure with 2 sister chromatids after the S phase. So the third stage is called the G2 stage, or gap 2, and here more growth happens. You can think of G for growth again, and this is the second one, so it's G2. And here the cell double checks everything. It makes sure that DNA replication's complete, it makes sure it's done accurately, and it makes sure it has enough resources for the 2 daughter cells. There's another important checkpoint here, and that's called the G2M checkpoint. This also involves proteins like P53 to make sure everything's good to go to continue. So interphase, which is composed of G1, S, G2, or just G1 and G0 in non-dividing cells, takes up most of the cell cycle time. If DNA damage is detected at any of these checkpoints, proteins like P53 can halt the cycle for repair or trigger apoptosis, in other words, programmed cell death, if damage is severe enough. The cell cycle is tightly regulated by internal signals, so these are primarily involving cyclins and cyclin-dependent kinases, which are also known as CDKs. So CDKs are enzymes that are only active when they're bound to specific cyclins. So the levels of cyclins will rise and fall through the cell cycle, and when they bind to their matching CDKs, they form an active complex, a cyclin-CDK complex, that can phosphorylate target proteins and push the cell into the next phase of the cycle. And once their job is done, the cyclins are tagged for degradation, and so that makes sure that the CDKs are inactivated until the next appropriate phase where they're needed. The cell cycle is also controlled by what are called tumor suppressor genes, and I mentioned P53, which is also a tumor suppressor gene. There are multiple types of cyclins and tumor suppressor genes, and they all work together to regulate when and how the cell divides. So when this control system breaks down, cells can start dividing uncontrollably, and that's essentially what cancer is. And it usually takes mutations in several key regulatory genes here to reach that point of cancer. And one major example is the TP53 gene, which encodes this P53 protein that we've been talking about. P53 is a critical tumor suppressor, and you can think of tumor suppressors as kind of the brakes in a car. They suppress tumors by stopping the cell cycle. They put the brakes on, and they say, wait, something's wrong, we're not going to continue. If these genes are mutated, it's like having a car with no brakes. On the other hand, you have proto-oncogenes, which are normal genes that promote healthy cell growth, but when they're mutated, they become what are called oncogenes, and these are more like the gas pedal. They're the ones that push the cell to divide. So an oncogene is more like a stuck gas pedal, whereas a mutated tumor suppressor gene is like not having the brakes working. So most cancers involve multiple mutations and usually both types of mutations because the cell has some kind of redundancy built in. But when you get this perfect combination of mutations, then you end up with cancers. Okay, let's move back into our mitosis process. So a cell that's going to divide after interphase, which remember that was G1, S, and G2, will enter what's called mitosis. And the goal from mitosis is to take one diploid cell with replicated chromosomes and to produce two genetically identical diploid daughter cells. This is essential for growth, repair, asexual reproduction, and there are 4 phases to mitosis, which you need to know. These are prophase, metaphase, anaphase, and telophase. And my favorite way of memorizing them is with the acronym PMAT, or Pass Me a Taco. Prophase, metaphase, anaphase, telophase. And I love tacos, like who doesn't love tacos? And so that one works great for me. PMAT, Pass Me a Taco. So first comes prophase. During this stage, the chromatin condenses into visible chromosomes. Each is made of 2 sister chromatids joined at the centromere. The nuclear envelope begins to break down, and the nucleus actually disappears. Meanwhile, the centrosomes move to opposite poles of the cell, and the mitotic spindle, which is made of microtubules, you should remember from the last episode, it starts to form between them. There are special proteins called kinetochores, and those assemble on the centromeres, and spindle fibers attach in preparation for chromosome movement. The next phase is called metaphase, and you can think M for middle. The chromosomes line up in a single file in the middle of the cell, on the center line, in an area called the metaphase plate. This stage includes a critical checkpoint known as the spindle checkpoint, or the M checkpoint, which ensures that all the chromosomes are properly attached to the spindle before the cell proceeds to anaphase. Then the cell will move into anaphase, and you can think A for apart. The centromeres split apart, and their sister chromatids are pulled apart towards the opposite poles of the cells by shortening kinetochore microtubules. So each chromatid is now considered an individual chromosome at this point. And then finally we enter telophase, and you can think T for 2 nuclei, where the chromosomes reach the poles and they begin to condense back into chromatin. A new nuclear envelope will form around each set of chromosomes, and the mitotic spindle will break down. And at this point, mitosis is now completed. So the 4 stages one more time, that's prophase, metaphase, anaphase, and telophase, or PMAT. Pass me a taco, because I'm hungry. So after mitosis, the cell will undergo cytokinesis, and this is division of the cytoplasm, which usually overlaps with telophase. And in animal cells, there's an actin contractile ring that will form a cleavage furrow, and it'll pinch the cell into 2 pieces. In plant cells, which are a little different, they have this rigid cell wall. We didn't talk about that, but they have a rigid cell wall on the outside of the cell, and cytokinesis occurs a little differently. There's a cell plate that forms in the center of the cell, and it grows outward until it separates the 2 new cells. And so either way, the final result is 2 distinct daughter cells that are each diploid and genetically identical to the parent. Now let's switch gears to meiosis. This is a special type of division that happens only in germline cells, and that's the cells within the gonads that are destined to produce gametes, which are either your sperm or your eggs. So meiosis has 2 goals. That's to first reduce the chromosome number by half. So you're going to go from a diploid cell to a haploid cell. And the other goal of meiosis is to generate genetic diversity. And so this involves 2 rounds of division instead of just 1. So you have meiosis 1 and meiosis 2. And this happens after 1 round of DNA replication during interphase. So it replicates the DNA once, then it divides twice, giving you 4 haploid gametes. The first part of that process is called meiosis 1. And remember, this is all after interphase in the cell cycle. So you still have the normal interphase that we talked about before, and then it's going to go into meiosis 1. And the meiosis 1 steps can be still remembered with the same acronym, Pass Me a Taco, because it has the same steps. There are just a few small caveats to these steps. The meiosis 1 prophase is a little bit longer and a little more complex than the mitotic prophase. It includes the usual prophase events plus 2 crucial events that are unique to meiosis. So the first unique event during prophase 1 is called synapsis. And this is where the homologous chromosomes, so 1 maternal and 1 paternal chromosome, pair up precisely. And so they line up gene for gene, forming what are called tetrads. Then they do something that's called crossing over or recombination. This is where segments of the DNA are exchanged between non-sister chromatids within the tetrad. So parts of the DNA from each parent gets exchanged to form new combinations of genetic material, and the points at which this crossing over occurs are called chiasmata. So this shuffling of genetic material creates new combinations of alleles on the chromosomes, and it's a major source of genetic variation in humans. If that explanation was at all confusing, I've got a figure for this in the notes, so please look over the figure in the notes if you're at all confused. But moving on, after prophase 1 comes metaphase 1. And here the homologous pairs, also called the tetrads, are going to line up on the metaphase plate. And this alignment is random, meaning the maternal or paternal chromosome from each pair can face either pole of the cell. So this is known as what's called independent assortment, and it's another major source of genetic variation, since it shuffles around which combinations of maternal and paternal chromosomes are going to end up in each gamete. And during this stage, the spindle fibers will attach to one chromosome from each homologous pair for when they get pulled apart. So just to go over that one more time, talking about the genetic diversity, during prophase 1, you have this recombination where maternal and paternal chromosomes can exchange DNA, and then during metaphase 1, you have this independent assortment where some of the maternal chromosomes will go into a certain side of the cell and some of the paternal ones will go, and so it's kind of random there as well. So the next phase is anaphase 1, and during this phase, the homologous chromosomes are pulled apart to the opposite poles of the cell. Remember, random assortment here. And importantly, the sister chromatids remain attached. So only the homologs separate at this stage. Then when you get to telophase 1, the separated chromosomes arrive at the opposite poles of the cell. This is followed by cytokinesis, which divides the cytoplasm and splits the cell into two haploid daughter cells. Although each daughter cell is now haploid, each chromosome still consists of two sister chromatids. So there might be a short interkinesis phase after meiosis 1, but it's important to remember that no DNA replication occurs before meiosis 2, since DNA was replicated one time already before meiosis 1. And so after meiosis 1, the daughter cells will enter meiosis 2, which separates sister chromatids. And so meiosis 2 looks very similar like regular mitosis, but the starting cells are haploid cells instead of diploid cells. It has prophase where the spindle forms. It has metaphase 2 where the individual chromosomes with two sister chromatids line up at the metaphase plate. It has anaphase 2 where centromeres split and the sister chromatids separate, moving to opposite poles. And then it has telophase 2, which is then followed by cytokinesis. The nuclei reform, chromosomes decondense, and the cytokinesis divides the cell. So that's basically the same as regular mitosis. So the final result of meiosis gives you 4 haploid cells that are each genetically distinct from the parent and from each other. So for meiosis, I would highly recommend looking at a figure. I tried my best to walk you through the process, but it's important to know how the chromosomes get separated and the ploidy of the daughter cells. So if anything was unclear, please look at the diagram that's with the show notes at reviewmcat.com. It's one of the things where a picture really is worth 1,000 words when it comes to following where the chromatids actually end up. And the MCAT also loves testing your ability to interpret a diagram like this, so make sure you can follow the ploidy and the chromosome number across both meiosis 1 and meiosis 2. Now, before we move on, I want to spend a few minutes just comparing meiosis and mitosis, since this is a key topic that's tested on the MCAT, and I want to make sure you get these questions right. So if you have the notes, there's also a figure outlining this like I mentioned, but we're going to quickly summarize some of the key differences. So as far as purpose goes, mitosis is for growth, repair, and asexual reproduction. And meiosis is for producing gametes for sexual reproduction and genetic diversity. For location, mitosis is in somatic cells and meiosis is in germline cells. Number of divisions, mitosis has 1 and meiosis has 2. Synapsis and crossing over happens in prophase 1 of meiosis, and it doesn't happen at all in mitosis. In mitosis, individual chromosomes align, and in meiosis 1, homologous pairs align. During anaphase separation, sister chromatids are separating in mitosis and meiosis 2, but homologous chromosomes are separating in meiosis 1. For mitosis, you start with a 2N parent cell and you end up with 2N daughter cells. In meiosis, you start with a 2N parent cell and you end up with 4N cells, or 1N cells. And for genetic outcome, mitosis yields identical daughter cells, where meiosis yields genetically distinct daughter cells. So that's kind of the comparison between the two I'm going to do. Meiosis produces genetic variation, and some of the key drivers of this variation that we mentioned already, but I'm going to say it one more time, is crossing over during prophase 1, which shuffles alleles between parental homologous chromosomes. There's independent assortment of homologous pairs during metaphase 1, and this creates different combinations of maternal and paternal chromosomes in the resulting gametes. And then a third source of variation that we didn't talk about happens during fertilization itself. So fertilization is basically random. So random fertilization means there's a random chance of any one of those unique sperm to fertilize any one unique egg. So this also creates a large number of potential unique zygote combinations. So I have a quick note here about errors during meiosis before we wrap up. Errors can and do occur during meiosis. One is called non-disjunction, and this happens when homologous chromosomes fail to separate during meiosis 1, or sister chromatids fail to separate in meiosis 2. And this can result in gametes with an abnormal number of chromosomes, which is a condition called aneuploidy. This can cause genetic disorders like Down syndrome, which is trisomy 21, or having 3 copies of chromosome 21. And I have one fun fact here. Plants can actually tolerate certain types of non-disjunction. And instead of causing problems, it actually leads to a larger, more robust fruits and flowers. For example, the seedless watermelon are actually triploid, which means they have 3 sets of chromosomes. And so this makes them sterile, but in watermelons, this is actually a bonus because you don't get those big hard seeds in the fruit. It's just these tiny little seeds that actually don't develop. So that's going to wrap it up pretty much for this episode. We'll do a quick recap of some key terms and key concepts here, and then we'll be done for meiosis and mitosis. So just to recap, the cell cycle includes interphase, which is G1, S, and G2, and that's for growth and DNA replication. This is regulated by checkpoints and cyclin CDKs. Cancer happens when this control gets lost. Mitosis occurs after interphase and has the steps with the acronym PMAT, or pass me a taco. That's prophase, metaphase, anaphase, and telophase. This makes 2 identical diploid somatic cells via separation of sister chromatids. Cytokinesis divides the cytoplasm by forming the cleavage furrow in animal cells or a cell plate in plant cells. Meiosis occurs in germ cells. It involves 2 divisions. Meiosis 1 separates homologs, and meiosis 2 separates sister chromatids. And this produces 4 unique haploid gametes. Meiosis generates genetic variation through crossing over, independent assortment, and random fertilization. And non-disjunction errors in meiosis will cause aneuploidy. The key differences between meiosis and mitosis, I'm not going to go over all those, but you should definitely go over those as a wrap-up. And this is all the blueprint for cell replication and gamete formation, which is fundamental to all life here. So that's going to wrap up this section of the series. Thanks for hanging in there with me. I hope you got as much out of it as I did making it. Be sure to come by the website, like I said, if you want more info or you want a copy of the show notes. It has the notes, diagrams, visual aids. There are also flashcards on the website for all of the shows that we've done so far. I'm going to keep putting those up as long as you guys want to use them. They're available on the website, and I've also got them on Quizlet. And you can also find other MCAT podcasts there as well. ReviewMCAT.com. Feel free to drop me a line, mark@reviewmcat.com. I'd love to hear from you, or if you have any issues or anything with the podcast. Like I always say, I try to do my best for it, but I do make some errors. If there is an error, I'm happy to correct them. And if you like the podcast, please leave me a review wherever you download your podcasts. So, in the next episode, we're going to move on to reproductive anatomy, hormones, sperm, eggs, talk about how those meiotic cell divisions contribute to building an organism. Thanks for listening to the MCAT Prep Pod Biology Review, and good luck studying. I'm hoping to review with you again in the next episode. So until next time.